Good morning, everyone. So we're going to have the workshop 5, Basic Science and Pathology 2. This session will be chaired by Dr. Wei from Beijing, China, Dr. Hikita from Osaka University, and Dr. Ninomiya from Tohoku University, and myself, Koga from Kurume University. The first presentation is intrahepatic IgA complex induces polarization of CAFs into the matrix phenotype in the tumor microenvironment of HCC. Dr. Som Pil-soo from Korea, please start your lecture.
Thank you for the kind introduction. I'm Dr. Song from the Catholic University of Korea. And today I want to talk about the role of intrahepatic IgA complex on the cancer-associated fibroblasts. It makes the phenotype changes in CAFs in the tumor microenvironment of HCC. CAF is a group of activated fibroblasts and have a critical role in tumor microenvironments. And there are many protein markers are reported to identify CAFs. For example, alpha smooth muscle actin, FAP, and FSP or S184 vimentin. And our group has recently were very interested in intrahepatic immunoglobulin A. Usually, IgA neutralizes pathogens against infections at the mucosal sites, for example, GI tract or respiratory tract. And IgA has also been reported to contribute to inflammation in dismantling in anti-tumor immunity in human liver. You can see the role of IgA in this picture. IgA binds to many kinds of cells, for example, monocytes, neutrophils, and bacterial surface, tumor cells, viruses. And these IgA neutralizes pathogens, but also transmits signals to these cells intracellularly. And our group last year have demonstrated the role of IgA complex on the intrahepatic monocytes. IgA complex binds to intrahepatic monocytes via CD89, the known IgA receptor, and the signal is mediated by YAP-TAZ signaling and PD-L1 expression and inflammatory phenotype changes in these monocytes dismantles anti-tumor immunity in HCC tumor microenvironments. So the paper was published in JITC last year by our group. And in this study, we tried to identify the role of IgA complex, not only on the monocytes, but also in cancer-associated fibroblasts in the TME of HCC. First, we collected patient samples, and these are the characteristics of patients' data. And in Korea, many patients of HCC are derived from HBV infection and also nephildio or alcohol. First, we counted the vimentin positive cells, which may be cancer-associated fibroblasts in tumor microenvironments of HCC. And we found out the positive correlation between vimentin positive cells and CD68 positive intrahepatic monocytes, and the negative correlation between CD3 positive T cells and vimentin positive cancer-associated fibroblasts. And these are representative immunohistochemical stain data from patients with high serum IgA and patients with low serum IgA and metastatic HCC in skin, there are no IgA. So in patients with high IgA and intrahepatic tumor, the vimentin positive cancer-associated fibroblast co-localizes with IgA in tumor microenvironment of these tumor. However, patients with low serum IgA, there are no IgA positive cells, although vimentin positive cells may be enriched in tumor microenvironment. And next, we performed single-cell RNA sequencing. we collected 3 patient samples by surgical specimens. The first patient was HBV-associated ACC with high serum IgA. And second patient, the HCC was derived from NASH, and the serum IgA was very, very high. And for this third patient, the patient was HCC was derived from NASH, but the IgA level was very low. In the first and second patients, there were a very high number of IgA positive, vimentin positive coughs in tumor microenvironment, and we subclustered in CAFs into 5 categories by single-cell RNA sequencing. And for the first patient, the second patient also, there were so many high number of IgA positive CAFs, and there were a very high number of C0 CAFs by single-cell RNA sequencing. These C0 CAFs were matrix phenotype CAFs. However, the patient with low IgA, the total number of CAFs were low, and there were almost none of matrix CAFs. So in this single-cell RNA data, we could hypothesize that patients with high IgA may have high number of C0 CAFs, meaning matrix CAFs. Next, we analyzed surgical tissue data by enzymatic dissociation of tumor cells, and we isolated CAFs and performed flow cytometry and also CAF culturing. And we found out that in patients with high serum IgA, there were increased number of FAP positive CAFs, meaning matrix CAFs by single-cell RNA sequencing. You can see that in patients with high serum IgA, the FA positive CAFs, there were a high number of FA positive CAFs, 5%, and these CAFs expressed IgA at a very high level. But in patients with a low serum IgA, there were a low number of FA positive CAFs, which do not express IgA. And we also found out that CD71, a transferrin receptor, also expressed in coughs. We identified the CD71 on the coughs by flow cytometry and Western blotting. And we also isolate coughs and we performed cough culture, and we treated the coughs with IgA serum complex. And after treating IgA in these cultured coughs, we identified that STAT3 signaling was upregulated, and the IL-6, which were downstream of STAT3 signaling, also the ELISA showed the higher level of IL-6 in IgA-treated calves. And PD-L1 expression also increased after treating calves with IgA. So IgA activates calves, and IL-6 level and PD-L1 surface expression upregulated by IgA treatment. And these PD-L1 upregulation mediates CD8 T cell attenuates activation of CD8 T cells, and co-culture experiments also demonstrated this decreased activation of CD8 T cells. You can see that by co-culturing by IgA-treated calves, the granzyme B and perforin expression, also TNF-alpha expression in activated CD8 T cells decreased by these calf co-culture. And this is the conclusion. Intrahepatic IgA induced polarization of ACC calves into more malignant FAP positive matrix phenotype, and these matrix calves upregulate PD-L1, and this PD-L1 upregulation decreases the cytotoxicity of CD8 T cells in tumor microenvironment of ACC. And our study uncovers 5 dynamic calf subpopulation and highlights their potential roles in tumor progression and immune suppression in HCC tumor microenvironment. Thank you for your listening.
Thank you very much, Dr. Sun. Um, Thank you very much, suggesting beautiful negative, maybe possible negative impact of IgA-treated calf on the immunological situation. So, may I ask one question? So, IgA has two types, monomer types and dimer types. So, if you treat the cell by IgA, which type?
Thank you for a very important question. And we treat calves with IgA with dimer type. Because monomer type cannot transmit signal into the cells. It only just neutralizes the pathogens, but the dimer type cross-links the receptors on the surface, for example, monocytes, neutrophils, and fibroblasts. So we treat the IgA dimer type.
But it is known that the serum IgA is monomer type. So
Yeah.
you show the high concentration of serum IgA may have a negative impact of the immune
Right.
situation.
But in the liver, the situation is different. In the liver, in the serum, IgA is mostly present as a monomer type, but in the liver, IgA is usually present as a dimer type because there are so many foreign antigens in the liver.
So it is related between the concentration of serum IgA and in lesional.
Right,
IgA.
because we have in the last year paper published in the JITC, we demonstrated patients with higher IgA have higher level of IgA in the liver, and that were positively correlated. By we performed assays in about 300 patients, and we demonstrated positive correlation.
Yeah. Dr. Arai.
Thank you for your beautiful data between the IgA and C0 calf. So I'm curious about the data because IgA-treated calf induced PD-L1 expression as well as PD-1 expression itself will be induced in cytotoxic T lymphocytes. So with this data, our hypothesis looks like the IgA higher patients can be better respond to the ICI therapy. Do you have any idea or speculation?
Okay, very critical question. But we have the data that patients with high serum IgA responds poorly. The response rate was very low by atezolizumab treatment.
Oh, really?
And we think that the PD-L1 expression in calves or monocytes are not very important. The inflammatory phenotype in the liver is important. You know that NASH-induced ACC responds poorly, irrespective of the PD-L1 status, because there are so many inflammation in the liver, and any PD-L1 treatment by atezolizumab may worsen the inflammation in the liver, so the tumor response may be worse. So we think the similar situation is between NASH-induced HCC and high IgA level in the liver, because PD-L1 expression here is not very important for atezolizumab treatment. And interestingly, in the last year paper by our group, we showed that patients with NASH-induced ACC show significantly higher level of IgA than patients with HBV-induced ACC.
Yes.
Yeah,
it's interesting. As widely known, the alcohol hepatitis patients have the tendency
very high.
to yeah. So did you also check the tissue or circulating the calf?
We have both checked. We both circulating IgA and tissue IgA. We checked double.
So are there any difference between the alcohol-related
positive correlation?
Oh,
when patients have higher IgA level, for example, alcohol-induced ACC, some patients have more than 1,000 IgA.
So C0 calf is also induced?
And IgA level in the liver is high.
Yeah.
And PD-L1 staining in monocytes and tumor-associated cancer-associated fibroblasts is unregulated in these patients. But the cytotoxicity by atezolizumab treatment is not very good.
Yes. Yeah. Thank you.
Yeah.
Are there any comments or questions? Yep. Thank you. Thank you very much for your exciting talks. Thank you.
It's my and the pleasure to invite the second speaker. The second speaker is the Dr. Jin Guangzhi from Tongren Hospital. Not here. Absent? Absent? In online? No? Oh, okay. Unfortunately, and Dr. Jin is unavailable here, and maybe later connected to us. So, and we switch to the third one. And third one is Dr. Naguya, and he will talk something for the interaction between an HSC and LSEC populations in the primarily gained environment of the treatment cytoglobin and knockout a mouse. Please.
Good morning. Thank you very much for your kind introduction. My name is Nguyễn Thị Hà, and I'm truly honored to be presenting my research today at the Apostle Oncology 2023. And the title of my report is Cell Crosstalk in the Microenvironment of DAT Nitrosamine-Induced ACC in cytokine knockout mice. We have no conflict of interest in this study. As you know, liver cancer is the sixth most common cancer worldwide, and ACC stands as a predominant form of liver cancers. And DAT nitrosamine is a carcinogen that has been used to induce ACC in mice for several decades. And ACC is induced by the ability of DAT nitrosamine to annihilate DNA and promote oxidative stress via the production of the reactive oxygen species that result in further DNA damage. And cytoglobin was the first member of the globin family in mammals, which were discovered in rat hepatic stellate cells. And we have reported that cytoglobin deficiency promotes liver cancer development from hepatosteatosis by activating the oxidative stress pathway, and cytoglobin attenuates pancreatic cancer growth via the scavenging reactive oxygen species. And the aim of the study is to investigate the cell-cell interaction in the pre-malignant and tumor microenvironment in cytokine knockout mice. And about the animal experiment, we have three groups of mice, wild-type knockout and transgenic, with 10 to 20 mice in each group, and liver cancer formation were induced by a single subcutaneous injection of DAT nitrosamine 6 microgram per gram body weight at 15 days old and observed for 12 months. And the primary non-parenchymal cells from both tumor and non-tumor part of knockout and wild-type mice were isolated, and high-quality cells were used for single-cell RNA sequencing, but transgenic mice were not included in the single-cell RNA sequence. For the principle of the single-cell RNA sequencing, the key to this technique is ability to generate tens of thousands of single-cell partitions. It contains an identifying barcode for downstream analysis. And for my analysis strategies, the main analysis contains cell clustering, cell type annotation, trajectory analysis, differential gene expression analysis, and cell-cell interaction analysis. And I show here the two packages that I use, SERAT and R package designed for QC analysis and exploration of single-cell data. And NICHNet modeling intercellular communication by linking ligands to target gene. And in the result of our animal experiment, we observed that both the number and the size of tumor increase in the knockout mice compared to the wild type, while they decrease in the transgenic. And TGF-beta1, TGF-beta3, which promote fibrosis and tissue remodeling, and AP1 transcription factor complex were upregulated in the knockouts. On the other hand, ATOP1, an antioxidant, were downregulated in the knockout mice. And ISC staining reveals the increased fibrosis and inflammation in knockout mice and decrease in transgenic mice compared to the wild type. And in the single-cell RNA sequence integrated data, we identify a total of 25 clusters, and we defining each cell type using the specific marker from the cell marker version 2 database. For example, CD68 is a marker for macrophage, which only expressed in the cluster 1. And LRAS is specific marker for stellate cell which expressed in cluster 4 and 7. And any cluster expressed less than 500 of genes are filtered out, and mitochondrial genes are high expressed in the cluster 2 indicate the cell damage were also filtered out. And finally, we annotated 10 cell types, including 2 subtypes of macrophage and 2 subtypes of stellate cell. And by D62 package, we further found 381 differential expression in the LSEC population between the DAT nitrosamine treated knockout and wild type. Notably, of all the significant different expression genes, selectin P were in the top 15 genes with a high git log2 for chain and lowest p-value, and under further investigation. By the cell-cell interaction and the circle plot here, so the top 20 interaction in the with the LSEC in the white high niche, and the quiescent stellate cell show only one ligand ATF strongly interact with the LSEC. Meanwhile, activated stellate cell so many interaction. We also interest in many ligand receptor pair. We need further analysis. Here we show you the expression of the AGF and its receptor, insulin receptor by quantitative RT-PCR, and AGF present is clearly found in the stellate cell, and its ligand were dominant in the LSEC. And in the knockout niche, both quiescent stellate cell and activated stellate cell have strong interaction with the LSEC. Among the ligand receptor pair of interest, SERPING1 was secreted from both quiescent and activate, and we have confirmed the expression of the SERPING1 and its receptor selectin P by quantitative RT-PCR, and the data from the own main cell type in the liver, so that the SERPING1 ligand were really expressed dominant in the stellate cell, especially from the knockout stellate cell, and its ligand selectin P expressed markedly in the LSEC and also white type stellate cells. And in conclusion, loss of cytokine promote liver formation in cytokine knockout mice compared to the wild type under the DAT nitrosamine treatment. And 10 cell types were annotated by single-cell RNA sequencing analysis. And there are 381 different gene expression in the LSEC population between the knockout and wild type mice. And numerous pair of ligand receptor interaction between stellate cell and LSEC are of interest and need further analysis. And thank you very much for your attention.
Thank you, very nice talk. And any question, any comments from the room? And maybe I can have the first question. So it's very nice talk and very well designed study. So did you have the more analysis for lockdown? If you have the wild type, lockout, lockdown, then you may find a dose-dependent reaction. So do you have the lockdown data?
Sorry, can you repeat the question again?
Okay, so you perform your study, use a lockout.
Yes.
Okay, and also you use the wild type as a control.
Yes.
Okay, so do you have data for lockdown to make the expression known, but still there?
Sorry, your question maybe is very interesting guessing for our next, analysis, but I will try my best to understand more for your question.
And maybe any other points. And did you have any data for the overexpression? So make the target gene into expression more and more.
The overexpression.
Yeah, overexpression.
Yes, we also generate the transient smile. It's the cytokine overexpressing model for the design, but we don't have the single cell analyze data for the overexpression yet. But in the next plan, we will add the transient, the overexpression into the analysis. Thank you.
So we'll have the data in future.
Yes.
Okay. So any more questions? Okay.
So may I ask? Excuse me.
Sorry.
May I ask a question?
Yeah.
One more.
Yeah.
Okay. So you perform the single cell analysis by using the knockout mice
Yes.
and then treated. So my question is whether if you drew the UMAP by single cell RNA sequence, so so you use the HSC specific knockout mice. So HSC population cluster were clearly differentiated different from knockout mice and wild type mice. So how about the UMAP data in your single cell RNA data?
Yes, actually, if I present all the data from knockout and wild type mice, it will different, but I use the integrated data. So it means that the cell type from the cell from the same cell type will clustering together. So it means the stem cell cluster here were divided by quiescent and activate, but it come both from the wild type and knockout mice.
So you mean the true mice and knockout mice and wild type mice HSC
Yes.
clearly differ in plot by using the subcluster analysis HSC cluster.
Yes, in separate data, but because we have 4 samples from tumor and non-tumor from wild type and knockout mice, so we need to integrate the data, combine the
I
data.
know.
Yeah, so if we separately analyze the stem cell from the wild type and knockout mice, there are a lot of different gene change.
Okay, thank you.
Oh, sorry, maybe I'm not understanding your question. Thank you very much.
Okay, thank you. And we will move to next week. And I will give the chair person to my colleagues.
So next presentation. Next presentation is anti-proliferative effects of flavonoid from anomine cysteine on hepatocellular carcinoma. Dr. Charpon Sangboming from Thailand. Could you start?
Thank you very much for a very kind introduction, and good morning, everybody. So I'm Jarupong Sangbunmi from Khon Kaen University, Thailand. And today I would like to thank the organizing committee that having me a chance to present my work today. And for this work, we don't have any conflict of interest. So let's start with just a briefly introduction about the liver cancer burden in Thailand. So in this figure, it shows you that the liver cancer has the highest incidence among all the people in Thailand comparing with the other kind of cancer. But actually for this graph, it includes both hepatocellular carcinoma and cholangiocarcinoma. And in our center is located in the northeastern part of Thailand. And as you can see here, the mortality rate of liver cancer in our area has really high comparing to the other area of Thailand. So that's why we are interested in the studying of hepatocellular carcinoma and also cholangiocarcinoma. And because our country is located in the tropical area, so that we have very abundant of the plant, the local herbal plant, so that we are trying to seek for the active compound that can be used as the complementary or the alternative treatment for liver cancer. And the plant that I'm going to show you today is the Anomianthus, and the local name is Nommelson. It's shown here, the flower of this plant that we used in our project. But actually for this plant, people in the local area, they use the decoction or the water extraction of the bark, the root, and also the stem to use as the galactogauge and also the antipyretic or to cure the fever. We are having a very good collaborations with these 2 professors in the Department of Chemistry, Faculty of Science, and then they did the extraction and isolation purification of the compound that we can extract it from the flower of this plant. And from the extraction and the identification, we can have the most abundant compound from this plant as shown here. 3 of them are in the flavonoid family, and 1 is the indole alkaloid. And because these compounds are very abundant, and then we think that it can be like the source for the bioactive compound that we may use for the anti-cancer aims. So we start with the screening of the effect into hepatocellular carcinoma cell line, HepG2 and Li7 or Li7 right here. And we found among those 4 extracted compounds, there are only one, pinosembrin, that show the cytotoxic for both cell lines that we have tested. And another one is chrysin that show like a mild effect on the proliferation of HepG2, but no effect at all in Li7. So that we think this compound might be the cell line specific effect, and then we just ignore that. So only the pinosembrin that which is the flavonoid, show the structure right here that we have selected for further investigation. And I just would like to inform you that actually pinosembrin is not a new compound. This compound can be extracted from the propolis from the beehive as well. And then we ask further what is or what are the mechanism underlying the anti-proliferation effect of this compound. We just did a simple experiment by using the flow cytometry. And we can see that the G1 phase of the cell cycle were arrested in both cell lines, as shown in this histogram and also in this graph. And then we prove for the proteins that regulate the G1 cell cycle phase, those are cyclin E, cyclin D1, and also CDK4/6. And it is interesting that this compound can inhibit the expression of the protein that control the cell cycle in both the cyclin part and the CDK partner, as shown in this Western blot. So we asked further, what are the upstream signaling that control the expression of these cell cycle regulatory proteins. So we have a collaboration with one of the staff in our department, Dr. Panupong Mahalabut. He helped us to do the molecular docking using the computational modeling to see whether which of the transcription factors that could be bound and then could be inhibited by the compound that we are interested. So we try to screen in several transcription factors, but just to make a long story short, I show you here, we compare the binding affinity and the binding energy between pinosembrin, which is the compound that we can isolate it from our plant sample, and with static, which is the specific inhibitor that is commercially available. So comparing the binding energy, we can see that pinosembrin has stronger binding to STAT3, much more than the known commercially available inhibitor of STAT3. So from this computer modeling, we prove further by doing the Western blot, and then we treat the pinosembrin into our cell line, which using the dosage that has the cytotoxic effect on the cells. And we can see that the phosphorylation of STAT3 significantly decreases when we treat the pinosembrin into our tested cell lines. And we have improved further by why the binding of this compound can inhibit the phosphorylation. But one of our speculation is that after the binding, probably it can change the conformational structure of STAT3 so that it inhibits the binding of the kinase that gonna phosphorylate this transcription factor. So in summary, for my presentation today, I would like to conclude that what we can find from our model is that when we treat the pinosembrin into hepatocellular carcinoma cell lines, it can inhibit the phosphorylation of STAT3, and then after the inhibition, it prevents the function of of STAT3 that are going to be like the transcription factors, so that it inhibits the expression of the cell cycle regulation proteins. Those are cyclin and CDK that I have shown. And finally, I would like to thank the research grant from the Fundamental Fund of Khon University, and also the travel grant from Thailand Tole Science Foundation, and also my research assistant and the team. And thank you very much for your attention.
Thank you, Dr. Champong. Is there any comment or any question from the floor? Okay, I have one question. This pinoseverin is one of the flavonoid family.
Yes.
So maybe I missed the slide. How about other flavonoid families? Does other flavonoid family have antiproliferative effects like pinoseverin?
The flavonoid?
Yeah,
Or, okay.
other flavonoids compound.
Yeah, usually people, they are interested in the flavonoid family because it can be like the antioxidants. And because we know the oxidant or the reactive oxygen species is involved in the progression of cancer cells, so that many or several of the flavonoid compounds, they show high toxicity to the cancer cells as well. So from this study, we just start with the plant that we know, and then we can isolate and we can purify it.
Okay, can we get to these plants another country like in Japan?
I'm not sure for Japan because I think it's a tropical plant. So it can be found abundantly in the Southeast Asia area. Yeah.
Okay, thank you.
May I ask a question?
Please.
So thank you, nice lectures. So you mentioned pinoseverin inhibit in STAT3 activation phosphorylation in your vitro data. So it's very interesting. So so have you ever checked some in mice model, for example, xenograft or orthotopic mice model or something like that model?
Yes, this question is very interesting, and we were asked by the reviewer as well. So from now, we haven't done in the mice yet. No, we don't have the animal model to prove whether it could be inhibit cancer cells in the xenograft. But we also plan for that.
I think so. And STAT3 in the tumor cell also act the tumor microenvironment. And also promote the HCC promotion. So I think that it's better to the better effect will be obtained in vivo model rather than in vitro model. This is just comment. So.
Yeah, thank you very much for your suggestion. Yes, I agree. I totally agree because STAT3 is one of the transcription factors that's involved in the inflammation process.
Yeah.
And then, as you suggest, if we can use the in vivo model, it will be like mimic the real situation in the tumor. Thank you very much.
And how about the AE in this adverse event? Do you have any adverse event information? Is this drug? So it is a toxicity? Do you have checked?
This compound or?
The pinoseverin and have so toxicity in the human?
I don't have that. I haven't have that information. Okay. I'm sorry about that.
Short question. Is this agent specific for STAT3?
Yes, for the one that we use is static, which claim is specific to STAT3, and it used to be tried in the clinical trial, but it has quite high toxicity, so that it stopped from the clinical experiment.
Another short question is the STAT3 is easily degraded. So did you check the degradation pathway or something about STAT3?
Pardon me, can you repeat the question
so,
again?
STAT3 is degraded by proteasome pathway. So you check the only the phosphorylation state. So did you check or are going to check the degradation pathway?
Yes, this one is also a very interesting point because from our Western blot, we can see just only the phosphorylation level are decreases, but the total form of STAT3 is still. So that we don't think the degradation going to be the effect from our compound.
Thank you.
Thank you very much.
Any question?
The final paper is the Professor Hashimoto. So the title is a protective effect of ischemic precondition on hepatic ischemic perfusion injury in rats. So please start.
Thank you very much. I make a presentation, protective effect of ischemic preconditioning on hepatic ischemia reperfusion injury in rat. Ischemic preconditioning, which means a short period of ischemia before ischemic reperfusion, was first reported by Murray in 1986 to be useful for myocardial protection. The same phenomenon has recently been observed in liver in animal experiment, and it has been reported that IPC increased the survival rate after hepatic ischemia reperfusion injury, reduced the spread of hepatic necrosis, and suppressed the elevation of liver enzyme. I suspect that many mechanisms are involved regarding IPC, but the details are still unclear. This study was to examine the impact of IPC on protection of hepatocyte after prolonged ischemia reperfusion injury. Method 24 Vista rat were randomized divided into ischemic precondition group, IPC, ischemia reperfusion group, IR group, and sham group. Hepatic ischemia and a traumatic clip was used to interrupt blood supply to the left lateral and median lobe of the liver for 90 minutes. IPC, liver was subjected to 10 minutes ischemia followed by 10 minutes reperfusion. 3 hours after liver reperfusion, all rats were sacrificed and blood and liver samples were collected. Model IPC group N8, 10 minutes ischemia, 10 minutes reperfusion. After 90 hepatic ischemia, 3 hours later, peripheral blood collection, ALT, AST, TNFR, MDA, HA. IR group, 90 minutes hepatic ischemia reperfusion, same 3 hours later, blood collection. Sham group. After 3 hours reperfusion, serum and liver tissue in each group were collected to detect the level serum ALT, AST, HA, hyaluronic acid, and TNF-alpha and MDA and liver histology. ALT IR group much higher compared to IPC group. AST also IR group much higher compared to IPC. TNF-alpha IR group is much higher compared to IPC group. Hyaluronic acid also the IR group much higher compared to IPC. MDA also IR group and IR group much higher compared to IPC. IPC group is good liver ALT, AST, and cytokine level, sinusoid, and MDA level. And histology, left IPC, right IR group. IPC group, good preservation of lobular architecture with less sinusoidal lining, endothelial cell swelling, and slabs of and few hepatocellular necrosis and neutrophil infiltration. But the IR group, disorderly liver sinusoid, enlarged and congested with many red blood cells, hepatocellular necrosis, and massive infiltration of neutrophil. PCNA labeling index, IPC group, labeling index 0.83 plus minus 0.06, but IR group labeling index 0.61 plus minus 0.04. IPC group is much better compared to IR group from the point of PCNA labeling index. Conclusion, ischemic precondition can protect the liver cell against injury. which may relate to cell proliferation during early ischemic reperfusion injury. Finish. Thank you very much.
Thank you for nice presentation. So you mentioned the precondition increase the cell proliferation and leading to the so better phenotypes. So comments and questions. So first, can I ask a first question? So I missed, or I might miss in your slide. So you preconditioned light lobe or total? Partial lobe.
Yes, sir.
So light lobe precondition and the so ischemic or also light lobe. So if the another lobe precondition, same effect were obtained or not? You guess?
Formerly the total ischemic and shunt, but partial clamp only. Most of other researcher the same model, therefore similar to same model.
So if the light lobe precondition and ischemic left lobe, so precondition effect is obtained or not?
Therefore, only right lobe only.
Right.
Therefore,
no study about this.
No study.
Okay, thank you.
But the mechanism is but the precondition is a good effect for the ischemic liver perfusion injury. Many researchers say that from the point of tunnel assay, many Caspar 3 and 8 assay. But in my data, I cannot check the tunnel and Caspar 6-3 and the Caspar 8. But the total data is ischemic preconditioning is good. In the future, much detailed mechanism from the point of tunnel and
but your ST and LT very dramatically decreased by okay. Dr. Miller.
Thank you for interesting research. So I'm Dr. Mura from Dutch Medical University. So I'm a physician, not surgeon. So please allow me the question from the non-specialist. So your precondition is a good result. So I think can we apply this precondition for human surgery in the future?
In our rat model, but in clinical, we use the precondition. Precondition is a good effect, but simple, no expensive, simple certain scale only. Therefore, the good effect for similar drug. Therefore, in our hospital, sometimes in case of liver resection, is conditioning method.
Okay, thank you very much.
Okay, all right, Doctor.
I'm Jun Arai from Aichi Medical University. Thank you for your beautiful data. I have two questions. So it's really interesting. Hyaluronic acid is upregulated after the ischemia. Hyaluronic acid. So what is the mechanism of that?
From the point of hyaluronic acid, hepatic sinusoid level reflex. Perhaps the ischemia preconditioning is good effect for sinusoid preservation of the sinusoid function. Therefore, the hyaluronic acid is the ischemic precondition group much lower compared to the control ischemia reperfusion injury. But I don't know the mechanism. Ischemia precondition is good effect for the preservation of the sinusoid. I don't know the our data, but the tunnel assay, according to the other researchers, from the point of tunnel assay, precondition group tunnel assay much decrease compared to IR group. Therefore, the apoptotic mechanism is good for the preservation of the sinusoidal function. I don't know the detail.
Yes, so in this trial, you performed the sacrifice, but if not, so if you check for longer time period, so if this model can be, the progress to the fibrogenesis, or it doesn't matter. So I'm curious about if you see more, much longer, 14 days or 28 days, are there any fibrogenesis or?
Yeah, good question. Only 3 hours later sacrifice, but the 1 day, 2 days, 3 days, 7 days after, if the animal arrives, check is important.
Thank you very much.
So, no question more. So thank you very much. Nice presentation. So unfortunately, Dr. Guanzi Jun is not online. So this session is closed. Thanks very much.
Thank you very much, chairs and lecturers.